Hi, this is Mr. Judd, and this is a blood smear. Some of the red blood cells in this picture are nice and round, and some of them have a crescent shape. The person that this blood smear came from had a disease called sickle cell anemia. Those sickle-celled shapes don't travel well through the bloodstream, and they can cause backups and clogs. They can lead to serious pain and even liver failure. In this video, we're going to talk about how a single change in DNA, called a mutation, can lead to major changes, or in some cases, no changes at all. Before we go too far, let's talk about what a mutation really is. A mutation is a change in the base sequence of DNA. Our keywords for today are point mutation, frameshift, karyotype, and chromosomal mutation. Well, what causes mutations? Let's start off with our classic double helix. Those rungs are the bases of the DNA ladder. Anything that causes a mutation is called a mutagen. A mutagen. Mutagens come in two basic forms: radiation. X-rays and ultraviolet radiation can strike DNA within the cell and alter one of the bases. The other major category are chemicals. Chemicals can attach to DNA and alter a base, and that can result in a mutation. The last category of change that could occur are simple mistakes. Mistakes specifically during DNA replication. If a mistake occurs during DNA replication and bases don't pair just right, that can lead to a change in DNA that can be passed on to future cells. The first type of mutation we'll talk about is point mutation. Point mutation is a single change in a single base within DNA. I have written down for you an original DNA sequence. It has 9 bases, 3 codons. Remember, it's read in groups of 3. DNA, remember, is transcribed into mRNA using the base pairing rule, and then it is made into a protein by the ribosome. And here are the amino acids. There are 3 amino acids in this small protein. Let's make a change. The change I want to make is I'm going to alter this base. I'm going to change it to a T. Let's see what that results in. The mutation is right here. We changed the G to a T, and that changed the mRNA. Look, it was a C there. Now it's an A because of that change. And because of that change, we have an entirely new amino acid. Instead of proline, we have threonine. So we have changed the entire protein with this mutation. And that protein may not function properly. It can lead to a genetic problem. There are also chances that when a DNA alteration occurs, when a mutation occurs, that it doesn't make any difference at all. If a base changes and that does not result in any change in the amino acid, it would be a silent mutation, one that doesn't really matter. Frameshift mutations are a little bit more serious. We are going to take our original DNA and we're going to alter the same base, but instead of replacing it, we are going to remove it completely. When DNA is read, it's read in codons, remember, 1, 2, 3, break. Where's the next 3? We just removed the G. The next 3 is GCT, break. We have changed the codons. Every codon down the line is now going to change. Let's see how that looks in the mutation. We have a new codon here. We have an incomplete codon here. That changes the mRNA, and that's going to change every single amino acid that comes after it. So a frameshift mutation changes a lot of amino acids within a protein. Now let's talk about chromosomal mutations. First of all, this picture is called a karyotype. Karyotypes are used to diagnose chromosomal disorders. They're a picture of the chromosomes within one cell. Here are the chromosomes of a normal male. How do I know it's a normal male? It's because there are 23 pairs of chromosomes, 46 in total, and the last pair are the gender chromosomes. It's XY. That's a male. If we had 2 X's, That would be a female. Let's take a look at this pair number 1. Remember, each pair consists of one chromosome from mom, one from dad. That's why they have pairs. And there are dark bars. These dark bars represent individual genes. If we were able to zoom in on those bars, we'd see the letters or the bases within DNA. If I take this entire leg of this chromosome and delete it, I have now deleted hundreds or even thousands of genes. Chromosomal mutations are very serious. One that is always mentioned in textbooks, and one that you might be familiar with, is when pair 21, instead of being a pair, is a triplet. If there are 3 chromosome number 21s, that results in Down's syndrome. Down's syndrome. N. Down's syndrome is the result of an extra chromosome in every single cell of the patient's body, and therefore they have way too many genes. And that means that gene expression, which we'll talk about in the next video, and the interaction within the proteins that are built are going to be altered, and they result in the symptoms you're familiar with Down's syndrome. There are other chromosomal diseases that will explore in class. To review, a point mutation is a single change in a single base within the DNA code. It can lead to a change in proteins, but it doesn't always. A frame shift is when one base is added or deleted, and then all the codons downstream change. A karyotype is a picture of the chromosomes within one cell, and they're used to diagnose chromosomal disorders or chromosomal mutations, where there's a change in one or more parts of a chromosome that can be added, deleted, or switched around.